I really want to congratulate Susan and her colleagues for running this series. Genetic literacy is really one of the most pressing social issues. If you think about the internet and cell phones and things like that have changed society dramatically, but unexpectedly, I think you can weigh the developments in genetics as being as powerful as those. And I guess the difference is that we're right in the middle of this wave of genetic transformation. And distressingly, as someone pointed out to me, most people don't get up in the morning thinking about DNA and chromosomes and things like that. So we actually have a long way to go to get there and to have everybody comfortable with genetics. Actually, I'm a little less worried about the whole thing this week than I was 2 weeks ago. I went to England, went to the UK, and you get off the aeroplane very early in the morning, and I went up to the customs officer or the immigration officer in England, and he said, "What are you here for?" And I said, a conference, a scientific conference. And he said, "What kind?" And I said, "Genetics." And he said, "Really? What's the clock gene?" So I said, and naturally I answered, "It's a gene that controls diurnal sleep patterns. It was discovered by my friend and colleague Joe Takahashi in the mid-90s in Chicago." And he said, "Well, that's pretty clever." He said, "What's the odd one out? T, C, G, A, or U?" I thought, I said, "It's just a quick trick question. I mean, one would think U was the odd one, but there's a lot of uracil in genomic DNA." I said, "This is pretty good, 20 questions at customs." And he said, "That's all I got, get out of here." But the message to me was that maybe genetic literacy is seeping out into unexpected places. So we're coming here to ask this question about should you have your genome sequenced, really as opposed to what terrible things could happen to you if you had your genome sequenced. And so let's consider that. There's a kind of a perfect storm of things that have come together to get us all to the position where we might actually ask that question. But I'm going to first divert into a very quick genetics lesson. So for those of you who this is old hat, please forgive me. And for those who are not, I hope you get something out of this basic genetic story. DNA, of course, is this miracle molecule with the 3 billion pairs of letters that come from each side of your family. So essentially each cell in your body contains 6 billion of these letters encoding the genes. There are about 20,000 of those genes. The genes themselves only occupy about 1% of all that DNA, and we don't know what a lot of that other DNA does. But essentially, all of your cells, with a few exceptions, contain a set of these letters that have come from your parents. So here's what happens during conception, fertilization, and procreation. You walk around with 2 copies of your genome. That's where you get the 6 billion, not the 3 billion. They're arranged into chromosomes, all of those letters, which are nicely coloured by a fancy technique here. And then during conception, you donate one of those copies, and your partner donates one of their copies, and so the new embryo, new fetus, gets back to 2 copies again, and there you have restored the full double complement of DNA. So this is kind of half of genetics. Now, I always saying in the foyer, I always tell people who come to visit my lab, you can get most of the principles of genetics in about 2 hours. You can get maybe 2/3 of them. The other 1/3 will take you 20, 30, 40 years. But the first 2/3 you can get it really quickly, as long as you believe that you and understand that you're getting most of the necessary information. So this is at the core of genetics. Person starts with 2 copies. They donate 1 copy of their genome with some reshuffling to a pool with someone else and form a baby. There you have for the next generation. Now, the next thing that makes it a little less simple than that is that sometimes the DNA doesn't stay the same, and there's this process of mutation. So mutation can occur about 1 in a million times that a letter is copied, which is actually a pretty high number when you think about it. And so every generation there'll be 200 to 500 changes in each of these genomes, of which, because the genes are only a small part of the genome, of which there'll be maybe 1 to 4 changes that actually affect a gene. So that's the sort of cumulative mutational burden per genome. So far that doesn't sound too bad either, 1 or 2 changes per generation. But the fact is that although a young species, we're some 10,000 generations extant since our original migration out of the sort of cradle of humanity. So these mutations, if they happen, have generation after generation to occur, you can start to build up quite a number. But here's the really sort of, in 2/3 of genetics, we're moving into the second third here. Here's the important point. if some of these mutations occur very early in the history of humanity, then probably in a room like this, then many of us are going to share those same changes. We'd be recognisable by those changes. But if some of those changes had occurred much more recently, then they might be more private to us. So if we went and mapped all those changes, we could actually calculate this frequency spectrum and use that to understand when these things had occurred in the history of humanity. So that's the basic message. The older mutations get to be more frequent out there in the population. Now, along the road to developing the current state of genomics, when we started to sequence individual people, we chose a particular character in genomics, Jim Watson, as one of our first sort of genome guinea pigs. And we chose him because he was, we judged, iconic in the history of DNA. It was Watson and Crick who discovered the structure of the double helix that Susan mentioned. And also because he was very competent at knowing what genetic information meant. It would be okay if his information was put out on the internet. And also he wasn't shy. He wouldn't mind talking about his experience. So we did that. We sequenced his genome in a collaboration with a commercial company. And there's my colleague Jim Lipsky talking to Jim Watson. Jim Lipsky is very used to giving genetic counselling services to families who are affected by particular genetic diseases. But here he is talking to a healthy 80-year-old man saying, I don't know, it's a lot of T's, A's and G's. That was his session. Now, in a less medical way, what we were able to discuss with Watson is that his first genome illuminated for us that actually there are a lot more of these kind of rare variants in him than we expected. So in this story of humanity coming out of Africa, where the older mutations will be more frequent in the population, and the ones that occurred more recently would be less frequent in the population, we found in Watson himself this unexpectedly large numbers of these kind of changes that we hadn't seen anywhere else. Actually, I'm twisting the story a little because we had our reference genome from the Human Genome Project, we had Watson, and we had Craig Ventnor's genome at the same time. It was really just among these three sources. But we saw more unique things in Watson than we expected. And this is the pattern that has continued and has been built upon as we sequence more and more people. We find that there are more of these variants that are rare and private than what we had previously expected. There's a person on an island, he's there because he has his own genetics that's really unique to him, not to anybody else. And what this means for a practical point of view is if we did indeed sequence all of you, there are many, many of the differences between you and the reference genome that you could talk about because you shared them, but we would find probably 100 to 300 changes in genes that you yourself had that no one else had ever had before. Can you imagine what this means for the physician who's going to be presented with people's genomes? People are going to walk in and show data, say, here's my genome, can you help me interpret it? There won't be anywhere to look up that has data that's quite the same. Now, the two tree pictures there are just a reflection of this is the population genetics. What's happened is that the human population, after it came out of Africa, was static for a long time and then grew very quickly. And it's this very quickly expansion and growth at the end that's allowed these emergence of these mutational changes that separate us all. But so that's the lesson in genetics, of which you now have 2/3 of human genetics. Okay, it's conception, diploids, babies get the copy from the mother and the father. There's mutation, and there's this variation in spectrum and changes that we can have. So back to our perfect storm. What are the ingredients that really have come together to bring us to this point of saying, should I get my genome sequenced? Well, these are the ones I list. There's this generally slow progress and improvements in medicine, and we don't need to delay that, but we'd love to see more diseases solved. Then there's this assumption of genetic determinism. So genetic determinism is really a concept that genetics matters, nature-nurture, that it's what you inherit makes you what you are. And there's a discussion about how true that is or how true that mustn't be. But in the context of disease biology, the genetics contributes a lot. If you understand the genetics, then you understand something about the disease. Then the other two ingredients for the storm are the Human Genome Project itself, which gave us this big reference panel to go look up and use in the computer. And more recently, the last thing there, the improvement of the actual technologies. Since we went from it costing a lot and taking a long time and to it now being much more facile. So let me just reflect on a couple of those points just a little bit more. The genetic determinism point, in the context of disease biology, it goes like this. Everybody knows that there are rare childhood diseases which have a strong genetic component, like muscular dystrophy, cystic fibrosis, etc. And so there'd be little. If you ask an audience like yourselves, usually, what fraction of things that would be bad health problems that may be lethal for you eventually have a genetic component, many people would say, well, it's a couple of percent because it's all these terrible childhood diseases that have, in many cases, poor outcomes. An audience who worked in the field might have a different answer. They might say, well, it's probably 60 or 70% because we know a lot of the adult diseases, although they are not strictly inherited strongly, they are familial. And if we do a calculation where we take people with the disease and people without the disease, and you ask how many of them had a primary relative with the disease, then we find it's higher in those cases that are affected. There is a propensity, if you have a relative with the disease, for you to be more likely to have it. That's called heritability. So there's a whole class, heart diseases, myocardial infarction, congestive failure, cancer, schizophrenia. All of these fit in this category of common disease and having a familial component. But then the more adventurous might go even further than that and say, you know what, I think practically everything's got a genetic component, except perhaps trauma. If you get hit by a bus, then you can't say that's genetic. Well, the retort for that, of course, is the thickness of your skull is genetic, and if you get hit by a bus, your chances of survival might be different. So if you want to stretch the analogy, then every single part of what you do, everything at MyQ is in fact genetic. So this genetic determinism in the context of a disease really is this pervasive notion that there's genetic underlyings for all the human maladies, and therefore if you understand those, you'll understand something about the human malady. Now, I won't say too much about the Human Genome Project itself. It is, as Susan mentioned, 10 years since we finished it, 13 years since we delivered this first draft. There's Francis Collins, who was the NIH leader of the project, with Craig Ventnor, who was the nemesis of the project. Some of you lived through this story. And Bill Clinton, enjoying himself, I think, having all the scientists there. And there's a big stack of phone books that is, if you printed the genome on them, then you'd have to fill all those books to represent the whole thing. One thing we did along the way with the Genome Project was adopt a position of free data release. Now, those of you who are scientists or who know scientists know that the idea of giving away your data as soon as you discover it is anathematic to the scientist. Most scientists never want to give it away until perhaps it's published and they want to display it prominently. So we all went to Bermuda, all of the representatives of the genome community, and we established this principle. We called it the Bermuda Accord. That is that sequence data would be different from other kinds of science data. It would be just put out there on the web for people to use straight away, and we would lower the barrier completely. There'd be no click-through license, there'd be no forms to fill out, there'd be no pre-qualifications. This data would just go out there. And this has been an important and resounding principle in the evolution of much of biology now, certainly all of genomics in the time since. And it makes a big difference to all of us because we all now can enjoy the access to these data in order to pursue our experiments and our entrepreneurialism and practically everything we do that relates to genomics. Can you imagine today if the internet had been not developed in the public sector? If there was some kind of a situation where every time you did an email you had to pay a penny, or if you wanted to start a business that used the internet, you would have to get a license to do it first? It sounds crazy, doesn't it? So genomics was headed to that direction, and it was this free data access that has allowed it not to happen. Now, from then and since then, we had this technical revolution, which is this last parts of this perfect storm. The cost for that original project was nominally $3 billion, and the sequencing portion of the Genome Project was probably about $600 million. And it took about 1,000 people all that 13 years. Now, with some of the new technologies and with the tweaks and perks of those, the cost has gone down to a couple of $1,000s per sample, and it all can be done in a couple of weeks. I've put here knowledge going from none to some. When we began to sequence the human genome, we didn't even know how many genes there were, and now we're getting an inkling of what a lot of those do. We're beginning to scratch the surface. And of course, the last thing here, the developments in computer technology, we already mentioned those and we all know them very well. So here we are in 2013 at the sort of end of this perfect storm with various ways that we can go and look at our own genomes. Now, this is the last bit of technology I'll subject you to, but there's actually different ways to do it, and it's essentially the difference between a very fine, high-resolution photograph, maybe a medium-resolution photograph, and then a photograph from 50,000 feet in the air. The picture on the left depicts the idea of having your whole genome sequenced, and this is what you can do for several $1,000s now. Get all the 6 billion letters, maybe you take some blood, you get the DNA, you sequence it, you get all 6 billion letters. You see all the 3 million differences that you're likely to have with the reference or with another person, and you can probably make some sense of the genes in there, the 1%. In the last few years, we've developed methods that allow you to zero in on the genic portions, on just the genes, and ignore a lot of that other stuff. This is a physical technique in a tube. You add things together and you mix things up and you pull out the genes physically, and then you just sequence those. So for about $500, you can look at just those 30 million letters and the 10,000 differences, not the 10,000 million differences, and the 1 to 300 unique gene changes that you have with a different person. That's the middle level resolution. And now many of you will know consumer companies like, direct-to-consumer genetics companies like 23andMe, where you can get another test for about $100. Who's had their 23andMe test done? Can we do a survey? A few people. So for a few $100s, you can get a test that just looks at about a couple of million individual letters that are known to be different in different people. These are the ones that are more commonly different in the population and can still give you a lot of information. So this is our 3 levels of resolution. So if you're a person thinking about getting your genome done, who would you be? What perspective would you be looking at the question from, and which of these kind of methods would you turn to? Well, these are the categories that describe everybody. I mean, I guess you can add some of your own, but you could probably also push those categories up underneath the other ones. So the most success that we're having right now is really in this area of individuals with genetic diseases and people who are considering having children who may have risk. Now, by success, I mean the most success in terms of demonstrating the power of science in medicine. So let's talk about those groups. We're able to demonstrate the methods were applicable to these kind of problems where a family might have a genetic disease which they knew was genetic because multiple family members had been affected and it had various other clinical characteristics that pointed straight to a genetic problem. We were able to demonstrate how these methods could solve the mystery of what the exact genetic lesions were. So after we did the Human Genome Project and got that reference sequence, and then we did the Jim Watson project that I mentioned, we turned to our colleague Jim Lipsky, who is the same Jim Lipsky who was counselling Watson in the previous slide, and a faculty member at the college. Jim has Charcot-Marie tooth disease, which is a peripheral neuropathy. CMT. It's a peripheral neuropathy. If you know Jim, you know that he walks with some difficulty because his ankles have been fused and his long nerves are not working like they should. So Jim is actually one of the world's experts on this disease because as it's in his family, and he's a clinical geneticist, he has worked to find the cause of that disease in patients. Now, when he started in his career, CMT was generally thought to be one thing. It was a syndrome, but it was basically one thing, with some clinical heterogeneity. But then Jim and colleagues found the primary cause of many of the patients, about 60%, I think is the right number, have a single kind of a change in the genome that leads to Charcot-Marie tooth disease. But it turns out there are at least 40 other genes that can be individually defective and give rise to the same syndrome. So there's a message right there. The syndrome itself is not one thing, it's at least these 40 different things because there's 40 different primary causes. Well, Jim had never found the cause in his family because he wasn't one of the more frequent causes, he was one of the rare ones. Even though he had his DNA and his brothers and sisters, and waiting there in the lab. So once these methods got to the right point, of course, we sent some surgery fellows from the Genome Center over to his lab and they bled him, brought back the DNA, we sequenced him, and in about a couple of weeks, and for a few $1,000s essentially, we solved the mystery of what was the cause of the disease in Jim Lipsky. So here's a picture of Jim. Down the bottom you see a pedigree that I think most of you are familiar with, but basically the squares are males and the circles are females, and the lines depict the family relationships. The dark filled in black squares are the affected individuals. So there are 8 Lipsky children in that generation, 4 had the disease. And the individual bad chromosomes bearing the defective gene tracked on the two sides of the family and showed that this was a recessive disorder where the two bad copies had to come together in him to give the disease. It was interesting that if we tracked those individual bad copies of the gene on their own in individuals who were heterozygous, who were carriers, just had one copy, they actually had subtle, distinguished clinical phenotypes as well. So it shows you the complexity of the genetics and how it can get interesting really quick. Now, this particular gene is involved in what's called endoplasmic reticulum recycling, so membranes inside cells and the way they behave. And so exactly what's going on there is really a big subject, of course, of Jim and others' interest, to see if there's one day a pill that can solve the problem of what's going on with that gene. So this was a demonstration that sequencing can be applied to a case of a genetic disease. Very useful for that family, for prognostics, diagnostics, and for further research, not yet for a clinical story. Soon after that, though, we had the good fortune to meet a family known as the Beery family, B-E-E-R-Y, and I have their website on the next slide, who had two wonderful children who, when we met them, were suffering from, they were twins, early teens, suffering from, one of them had very bad drooling and shaking, and the other, she suffered from bad breathing episodes. It had died almost a couple of times with the emergency team coming to the house to resuscitate her. The family was very distressed. Their longer history was they'd been born normally, they'd had some early childhood problems. They were actually diagnosed as having cerebral palsy, which is kind of a catch-all for this kind of a problem. The mother, wisely, and through reading the literature, recognised they may have a dopamine deficiency. And a disease called Sagawa's disease had given them dopamine supplements which restored much of their problems, their normality as children. But in the early teens they reverted and they were suffering from some quite other difficult problems. So when we saw them, they were really quite distressed. The family was in a bad state. They'd been on what we call this diagnostic odyssey. That is where the parent takes the child to every neurologist in the country because you never know when you're going to find the one person who can really understand what's going on here. We sequenced the children and quickly found it was actually a gene that's not just involved in the dopamine pathway, it's also involved in the serotonin pathway. So as the father said to us in the genetic counselling session, oh, you mean we only had half the story? The answer really, from the scientists at least, was yeah, that's exactly right. You only had half the story. Their own neurologist put them on the additional supplements for the serotonin supplement, and literally within weeks, they are restored. The shaking went away, the drooling went away, and you can see they're beautiful. This is a film star family. They're just wonderful people, and the children are winning high jumps and sports events and just doing tremendously well. So this is our poster child for the case where this family needed genome sequencing. Genome sequencing led to a definitive diagnosis of their condition, and also for a therapy that restored them into a really good state. My slides seem to be on automatic, which is good for you because it means I'll get through them in a nice steady pace. So we were, and the college, of course, we were buoyed by this, by all those other elements of the perfect storm, and the developments in the technology. And together, the Genome Center and the Department of Genetics launched a new laboratory that focused on sequencing all the genes in children which had suspected genetic conditions. And you see from the growth in the curve here, we started nearly 2 years ago now with just a handful of samples from well-connected physicians who we were able to encourage to send them, to pretty much a steady 200 samples a month now. And so we've converted this from a laboratory research exercise with some stunt sequencing of ideal candidates to something that's pretty routine. The cost, the pre-insurance cost is quite high. It's around $7,000. That includes much of the clinical contact and validation and all of that, and we're working to bring that down. The speed is a couple of months. We're working to bring that down. It's a steady test now, and it returns a result of a definitive diagnosis in these children about 25% of the time. As the family comes in, not sure what the problem is, about 25% gets solved. The remainder, we're ushering those individuals, giving them the opportunity to participate in research protocols so that we can use other methods to come in and find out what the cause of their problem is. Now, this group is attended to by the entire Department of Genetics. clinical sign-out group who looks over the data and discusses each case and looks up databases and decides exactly what the genetics data mean, because at this time there is no single computer algorithm where you can take a person's genome and put it in a computer, push the button, and get out a genetic story of the person, unless things have been seen before. But the point of my long story earlier about coming out of Africa and now we find this unexpected number of differences between us is, more often than not, you see things that no one's ever seen before. Okay. These clinics are seeing a lot of new changes in genes for which something is known, but not a whole lot. Anyway, the family gets a report that looks something like this, and I chose this particular one because it's actually mine, and I have, after a long period of time of resisting, gotten my genome sequenced. So I can put my hand up if we say, who's had their genome sequenced? Okay, so we're in a less and less exclusive club as time goes by. Well, I'm happy to tell you my genome is really dull. I'm a carrier for one disorder which there's not in, I have one child and my child's healthy, doesn't have this disorder. And that's what gets returned to me, the carrier status. If I was there with the child, this was the child's genome, and there was a genetic disorder, then the result, if positive, would indicate the gene, what other evidence there was about that particular change in the gene, and what that would likely mean clinically. Also, what is reported is what's called incidental findings. If there's a finding of a gene which may have implications for the family, then a change in a gene that has implications for the family, then that too is reported back to the referring physician. So this is a very controversial part of this whole process, because if you send a child in for a metabolic disorder for a genome sequence and you find a breast cancer gene, that's important not just for the child but for the parent. So the parent has to decide what to do about that. This is at the nub of these problems. There's a few other things there I'll just mention again in a moment. So one of the I guess byproducts of doing this, and perhaps what we'll be able to do when the tests are truly ubiquitous and more extensive, is to apply this for simply carrier disease testing. So I think you're probably mostly familiar with the idea of carrier disease testing. That is, you screen a person to ask if they have a change in a gene, if they meet a partner who has also one bad copy of that gene. You both have one normal, one defective copy of the gene. If you have a child, you have a 1 in 4 chance that you'll have 2 copies and you've got a problem. This is classic cystic fibrosis, etc. So a sort of byproduct of this report is the ability to do this carrier testing. So another controversial question we've got to face is, what's the real value of comprehensive carrier testing, since we now can look at about 2,000 genes, about a quarter. The number of children with a problem is probably around 1%. If you say what's the frequency of genetic disease in children, it's a little bit higher than 1%. Probably about half of those it's really genetic, half it's something else. And amongst those that are really genetic, there's probably a third of those have a recessive disorder. So something like 0.25% of live births might be informable by a carrier test in parents, if you follow that logic. And the question that's controversial, of course, is what you do, and we won't discuss that here tonight, but of course there is risk awareness, there's the options for termination, for managed fertility in the future, and there's also looming a process we'll call non-invasive early diagnosis, where it's possible that the mother could be, through serum sampling, plasma sampling, very early in pregnancy, monitored quickly and inexpensively to determine the status of the fetus. Let's leave all that there, because I don't want to really hold the conversation on that very difficult set of topics. So back to who are you and who would you want your genome sequenced? So if you're not a person with a genetic disease in the family, and you're not actually about to have children, who else might you be? Well, you may be an unfortunate person to have cancer. And the question comes, shouldn't I have my cancer sequenced? There is a reasonable set of thinking that says that maybe cancer will actually drive this whole enterprise of genome sequencing, because there's so much cancer around that everybody should have their cancer sequenced. So here in our camp, we've been saying actually genetic disease is probably the first one because it's more tractable. Cancer sequencing is actually very difficult and the yield is low right now. But nevertheless, there is certainly a place for cancer sequencing. The logic goes something like this. This is an illustration of a cancer. That's a particularly nasty one. It's a glioblastoma. And you can ask yourself what's going on in that disease. Well, here's what's happened. Here's you starting off as a single cell at birth. And that cell, of course, divides and forms many, but the progeny of that cell will go through childhood, through adulthood, and then through some pre-cancerous stage to a cancer in an individual who develops malignancy. After the malignancy, there'll be some kind of treatment and then potentially a relapse. But you can trace the fate of a single cell like that over time. And what can happen is that there may be an inherited mutation that has come into that cell. You've got it from your parents. It's a predisposition to the cancer. Maybe, maybe not. In addition, there's the opportunity for changes that occur to that cell during its lifetime in you as a single person. This is called a somatic mutation. It can be spontaneous, but it can also be, the frequency can be boosted a lot by environmental effects like chemicals and smoking. And so a mature tumour can have many, many such mutations that can be discovered by taking a sample of the tumour and sequencing its genome, and then taking other cells from that patient and sequencing those and then comparing the two. And the difference may be from tens to thousands of changes, but amongst those there may be key changes that are signatures of that cancer and that are relevant for the therapy of that cancer. Now, that's the whole logic of cancer sequencing, driving discovery and driving treatment. The status right now is, I think most would agree, somewhat embryonic. That is, you can reduce the number of instances of known changes in cancer genes down to a relatively small number, such that you would probably prefer to do some specific targeted test rather than sequencing the whole cancer genome to find those changes. In about maybe 5%, it's a back-of-the-envelope calculation of cases, would a comprehensive test or a rapid screen test that's more focused give you information that can lead to help with treatment. In many of the cases, other kinds of tests, non-DNA tests, can give you the same answer. Of course, the classic one is Herceptin, where you don't need a DNA test to look for that kind of treatment in breast cancer. But the future of this is bright. The idea, and the diagram in the bottom right, is meant to simply illustrate the complexity of the biochemical pathways that can be disrupted in cancer. And if we can get to the point where a whole genome test can interrogate those entire pathways and tell us on an individual cancer basis what kinds of changes have happened, this will lead to new avenues for cancer treatment, and with new drugs we'll have new matches and we'll boost that number from 5% to much higher in cancer therapy. So if you're in neither of those categories, which is ideally most of us, if you're a healthy adult or you're just curious, what would happen if you went to get your genome sequenced? Well, as I intimated just before, I'm one of those people now who can stand on the stage and say, hey, my genome's sequenced, and can answer to that. Actually, I've got to tell you, parenthetically, this is the second time I've had my genome sequenced. I had it sequenced back when we sequenced Jim Lipsky, but it turned out that his was so much more interesting. He got his picture in the New York Times, and we got a paper in the New England Journal of Medicine, whereas I have almost no alleles that anybody cares about. So he really got all the attention. But the technology was an older technology, and so with the newer methods, there's actually new findings. And of course, that first data was from 4 or 5 years ago. Now there's a lot more data generated that allow us to do much better interpretation. So here's my part of my family story. That's me and the little arrow in the bottom right. But my mother had Waardenburg syndrome, which is actually pretty interesting. It's heterochromia. If you've ever seen anybody with two different coloured eyes, it's kind of spooky. And a little shock of white hair and deafness. She was really quite deaf. Now my hearing's not that great, so my frequency range is starting to go, and I've kind of been wondering if it's a genetic thing or not. So I was actually looking at this data mainly with the interest of seeing if I could find something there. So I have here a report, which is not actually the report that gets the clinician. You can't read it here. I'm just showing the complexity of it. But if we zeroed in on the top line, you'd see I have a carrier for Wolfman syndrome. And this disorder does carry some hearing anomalies in the individuals who are affected by it. The mutation I have, I think, is a more benign one. It is an autosomal dominant condition, but the gene change I have is probably not a bad change for the gene. But this, for me, is my hint about maybe this is something to do with my hearing. So this is just introducing you to what you can do when you get your genome and you start to fossick through it. Here's another part of the report. These were on the earlier report. These are some of those markers that are to do with drug metabolism, with warfarin and various other chemicals. You can see I've got this one that means that I don't metabolize nicotine very well, and various other things like that. and probably I'd need a big dose of warfarin if I had to take it compared to some other people. So this is actually what we call genome gazing. So for a very, and some other words for it. Of course, like all of you, I'm kind of interested in where I came from, but this now is back to the 23andMe. I've just taken this from the 23andMe page. Unfortunately, if you go to my genome, it is really pretty dull. I'm almost entirely European, and with a little British and Irish, and just frankly nothing particularly exotic in there at all. So I had to turn to a friend and an ex-student. I called her up. I said, "For heaven's sake, send me your 23andMe picture so people have something interesting to look at." And so she's originally from Mexico, and you can see it's a much prettier picture. And similarly, I asked another friend, I haven't got the slides, who's half African American and half Brazilian. So pictures can get much better. Here's another view too. And here's the point. Really, the serious point to make here is that these data at the whole genome level can very, very precisely predict your ancestry. And to the extent there's data out there, can predict your ancestry and indicate where you're from. These tests are done with a very low resolution example of genome interrogation, and they still make it look like a parlor trick. So it's pretty easy. Now, if you follow the literature on all of these notions in the last year or two, you'll see that amongst the scientific community, there are a few people who have got up and sequenced their own genomes and have talked about how it has changed their health history and what they have done. Their knowledge has led them to have a different view of their own health and to pursue different clinical outcomes. So I'd invite you to look at Mike Snyder's story. Mike's the chair of genetics and a friend and colleague, and he sequenced himself and decided that was the reason he was declared a diabetic. It's an interesting social thing, because some of us who are familiar with the locus that he points to and the more technical issues of the story would say that maybe he wasn't so much prompted by the knowledge of the gene he found, but just by the process he was going through. He had a subsequent glucose tolerance test, which was actually informative for him about his way to handle glucose, but the genetic data itself was a little bit indirect. Nevertheless, I was at a social situation recently, and someone told me Mike's story and said how great it was that genome sequencing was being used. So I felt conflicted. On the one hand, this is terrific. This is what we need to get genome stories out there. And yet, on the other hand, the technical underpinnings could be called into some question. Anyway, to make Mike not feel good about it, we dubbed this the narcissome. That's what you are if you stare at your genome too. So who else should be asking themselves these questions? Who else should be looking for genome data? Well, there's sort of this boundary of the healthy individual and the researcher and the altruist. And many people have committed their genetic data into research studies, and the world is much better off for it. And normally myself and others in the field spend all their times at lectures talking about those kind of instances. Here's just one little snippet. It's that if you take that graph there, it shows the triglyceride distribution in a randomly selected group of people. We have a program where we've sequenced about 10,000 of those people. And if you look through them, and you look with the kind of eye for looking for complicated genetic stories, you're stunned to see that a few of those people are out on the edge of the distribution because they have single gene defects that lead to the problem. So you begin to understand how single gene changes can contribute to the overall genetic architecture of these adult diseases through the sequencing. So the contribution to science through making your genome available has been a wonderful thing. Now, we're going to see a shift in the need for people to do that. I mean, right now, there's a chance that you personally would have been involved in a study, and if you read the consent forms they're giving you, then you might find that you're being asked to donate your genome so it can be sequenced and maybe even put out there on the internet like Jim Watson. There's probably going to be a lesser need for that because we're seeing this progression into this clinical regular use of the data. So you see, I'll depict this in this diagram. You see that big blue circle on the left. That's kind of the state of play right now, or maybe the circle in the middle, the blue one, where most of these kind of things are being done in a research arena, either on its own, where people go out and find key individuals for sequencing, or else it's been done near a clinic, and after that research activity, samples are pushed over into the clinical, into that red circle, into the clinical medical record. But like X-rays and CAT scans and all that kind of thing, as this gets more and more frequently applied, and as the cost goes down even a little more, we're going to see more often that the call for the sequencing is simply for a clinical test. And after the clinical test is done, the data is going to get put into the medical record. And there it'll be, and then somebody will come along and say, well, I'm a scientist, I want to study diabetes. That person will no longer be out there looking for people who have diabetes to ask, may I have your blood for a study? They'll go simply to the clinical records and say, let's look at those people with a certain clinical background, then ask what's in their genomes. So this is the societal shift we're going to see in this area. But in the meantime, we've got to ask the altruists to please go and look at their data. So this illustrates that it's going to be big. So if you do that, and you in the research arena where you're not so protected automatically by these clinical restrictions at looking at your data, what are the kind of risk scenarios that you like to talk about? We love to talk about these kind of risk problems. There's really two views of this, sort of stepping back for a moment. There's the view that these are real risks, that they're important, that we all should think about them, and that we should work out genetics as it grows in our lives in the context of these risk scenarios. The other completely opposite view is that people used to, in certain primitive tribes, used to think the same way about photographs. You know the story. If you take the photograph of the person, you've somehow captured their soul or some part of them. Maybe that's how we'll look at our contemporary views of genetics as time goes by. That illustrates really the full diversity of opinions in the community. But here's really the risks all in one slide. There's the bad news, that is, if you go and donate your genome into a clinical test, it may be that you found to have an unhealthy allele that you don't want to know about. There's a very small percentage of that likelihood of that happening, but I can tell you it does happen. The second thing is your genome, of course, is not just yours. It's your children's, it's your family's, it's your relatives, alive and dead. So there's a risk that you confer, or a knowledge that you access, if you tell everybody about it, that is then accessed by and for and behalf of your relatives. Okay, that's something to think about too. On the other side of the coin, how is this relative to other privacy? There's the argument that, hey, nothing's private anymore. The NSA should just leave our telephones alone and go look at our genome sequences. And then there are three sort of prototypical stories here that all have to be told. The first one's the sperm donor story. So this story is probably about 8 years old. It's about a child who decided they wanted to find their natural biological father. And the only information that they had was the county that the father was from, and there was date of birth of the father. Okay. So that still left a lot of people. Well, with a minimal amount of DNA sequence information, the child went to Ancestry.com or one of the other direct-to-consumer suppliers, and with a minimal amount of sequence matched that database and found a family name, a small number of family names with a high likelihood of a match. That data plus the demographic data nailed the father straight away. So that was an illustration of how, once you do this triangulation, how powerful the data are. The cocaine control story is hypothetical. It's this idea. Supposing that you, in your altruistic mode, you go, supposing your cousin in their altruistic mode decides to go and be a control in a cocaine addiction study. So let's have the people who are addicted to cocaine and the people who have never tried it or have some other reason to say they're not addicted, put their DNAs in. Let's have all that data on the internet and available for review in an uncontrolled fashion. Then you go to get a job and you have some of your sequence associated with your application for a job. Of course, you come up with a high likelihood of being a hit on the database that contains all these people in the cocaine study. So that ruins your employment chances. That's a scenario. And the final one is the crime scene story. This is so the facts here are, of course, people finding. Of course, the scenario where there's the perpetrator is nailed because we finally find the DNA. But there's now the examples where the DNA is found, and then the relative of the perpetrator is identified, and the local environment has the actual perpetrator, and the police find that person through this indirect connection. So the scenario here is, what if your 16-year-old child smokes marijuana and then throws the thing away, and then somebody picks it up and gets the sequence from it. Bang! That's the crime scene story. So there's plenty of look out things there for us all to consider as we go into the risk scenarios. So how do we add all this up, if we sort of pull this together? It's pretty simple, really. I think if you have a genetic risk, and if you have a family, if you're thinking of procreating, and you have a concern about anything genetic, or if you're particularly curious or altruistic, or you're narcissistic, I guess I should have just put up there, then really you should have gone ahead, you should really go ahead and consider looking at some of these data for yourself. If you're concerned about privacy, if you just genuinely uninterested and you're a healthy adult, I don't see a single reason that you should go and get your genome sequenced, even though I personally would advocate everybody, I'd like everybody to. There's no reason for you to do that. But if you take that course of action, I think you should prepare yourself that soon, whether or not it's because of a medical emergency you didn't anticipate, maybe a suspected malignancy, or even something as benign in the future as going to the dentist, you'll end up having a genome sequence and put somewhere in the record, so maybe you should prepare yourself. So that's all the formal comments I want to make. I hope we got some good time for discussion. I do want to thank the team that I work with, and I've got David Wheeler is here in the audience. He runs the cancer program in the Genome Center. I see Sam Penny somewhere. I think she stepped out, one of the genetic counsellors. But the individuals up here who run the program down at the college, and especially want to thank Susan and the other people who put this program together for tackling genetic literacy. Thank you.